Good morning, children. So, we have been introducing into a new pathway, uh, that is the de novo pathway of fatty acid, the biosynthesis of fatty acids. So, the fatty acids can be synthesized in the body or can be present in the body from various sources, like the dietary sources that we have already discussed during the digestion. So, that is one route through which the fatty acids can be present in the body. Another source is that it can be synthesized as a result of a breakdown of the depot fat. Right, when there is energy demand, what happens? The TG undergoes breakdown. TG will be mobilized from the adipose tissues and they will be taken into the liver. They will be undergoing TG breakdown, whereby we can have the fatty acids. There is yet another source, that is the body itself can synthesize fatty acids from the excess quantities or surplus amounts of carbohydrates and proteins. That is, if your diet is having more and more amounts of carbohydrates, the calorie has to be utilized. The calories thus obtained has to be utilized, and the calories are stored in the form of fatty acids. Clear? So we have explained the various route through which the fatty acids can be entered. Right? So it can be from, that is glucose, which is present in the carbohydrates, will be converted into pyruvate, and this pyruvate is from a diet, then can be by adipolysis. That is, the food can have carbohydrates and proteins. These carbohydrates and proteins will be undergoing conversion to give rise to acetyl-CoA, and this acetyl-CoA undergoes condensation reaction, which is almost the reverse of the fatty acid breakdown. Beta oxidation, the reversal is happening during the fatty acid biosynthesis. So, which are the sites where the biosynthesis happens? It can happen in all the tissues because it is a purely cytosolic process, right? So, the excess carbohydrates and proteins will be converted into a useful form of substances, which is known as the acetyl-CoA. So, these acetyl-CoA can condense with each other almost in a reverse fashion to give rise to the biomolecule fatty acids. So, this pathway through which the fatty acids are synthesized from fresh raw materials is known as the de novo pathway. It is a purely cytosolic process, and hence the enzymes involved are also present within the cytoplasm. But the problem is that the acetyl-CoA formation from the carbohydrates or proteins, whatever it is, whatever be the source, the acetyl-CoA or the pyruvate, the acetyl-CoA is synthesized primarily in the mitochondria. So from the mitochondria, it has to be taken into the cytoplasm, and it's not that easy. It's a tedious process, and hence in order to translocate the acetyl-CoA from the mitochondria into the cytoplasm, there is a transport mechanism required, or shuttle system required, that we have already studied. That is a malate shuttle system, right? Yes, so this is a malate shuttle system. So acetyl-CoA itself cannot cross the inner mitochondrial membrane. So what happens? Acetyl-CoA first condenses together with oxaloacetate in presence of citrate synthase enzyme to give rise to citrate. The citrate can cross the IMM. So the citrate will be translocated from the mitochondrial matrix into the cytoplasm across the mitochondrial membrane, IMM and OAMM, and finally it reaches the cytoplasm, and on entering into the cytoplasm, citrate will be cleaved to give back the acetyl-CoA and oxaloacetate by the enzyme ATP citrate lyase. So ATP citrate lyase, in presence of one molecule of ATP, by consuming one molecule of ATP, citrate, the 6-carbon citrate will be cleaved up to give the 2-carbon acetyl-CoA and 4-carbon oxaloacetic acid. So this is how the acetyl-CoA is generated for synthesizing the fatty acid in the cytoplasm, right? Now what happens to the oxaloacetate? The oxaloacetate has to be taken back into the mitochondria because our TCA cycle has to be carried on, right? So oxaloacetate will be moving towards the mitochondria. Again, the problem is that it cannot itself cross the membrane. So oxaloacetate will be first converted into the malate, which is a transportable form. So oxaloacetate malate conversion is carried out by MDH, malate dehydrogenase, by consuming one molecule of NADH. So oxaloacetate is converted to malate. Malate itself can cross the IMM and can reach the mitochondrial matrix, or there is no other alternative route that malate can be converted into pyruvate so that NADPH is produced. Again, this route is preferred route because the fatty acid biosynthesis require NADPH as a coenzyme, right? So this NADPH is obtained from the conversion of malate to pyruvate by the enzyme malic enzyme. So it's a reductive biosynthetic reaction through which malate undergoes oxidation to give rise to the keto acid pyruvic acid, right? This pyruvate can enter back into the mitochondria and can enter can be converted into oxaloacetate by pyruvate carboxylase, and hence oxaloacetate can be regenerated and the TCA cycle can run, right? So this is how the raw materials are obtained. The acetyl-CoA now we are having and the NADPH molecule also. But the reaction is an energy requiring reaction. It consumes one molecule of ATP. Now the acetyl-CoA is within the cytoplasm. So this forms the stage 1 of fatty acid biosynthesis, right? So fatty acid biosynthesis requires 3 different stages, or the pathway involves 3 steps, 3 stages. The first stage is the production of the 2-carbon moiety acetyl-CoA, or the translocation of acetyl-CoA, the 2-carbon moiety acetyl-CoA from the mitochondrial matrix into the cytoplasm with the help of malate shuttle. Now second stage is the carboxylation of acetyl-CoA. That is, acetyl-CoA undergoes undergoes the formation of a next higher member of the family, which is known as malonyl CoA. Because what is the purpose of this carboxylation reaction? The fatty acid biosynthesis is a series of condensation reactions. That is, 2 carbon moieties are getting added up to give rise to finally a C16 fatty acid, which is a palmitic acid. So it's a condensation reaction. CC covalent bond is getting condensed together. So naturally, the reaction is a high energy reaction. That it is thermodynamically highly demanding reaction. Thermodynamically, it is not that easy for the reaction to take place because 2 carbon atoms are getting joined together, getting clubbed together by means of the formation of a covalent bond. So it's a thermodynamically, it is a very tedious process. So the reaction cannot occur by itself, right? So this is made much more easier by converting one molecule of acetyl-CoA into its next higher member, which is the malonyl CoA. Otherwise, the reaction is thermodynamically unfavorable in nature. No 2 acetyl-CoA molecules can directly come and get added up. So what is the alternative? The one molecule of acetyl-CoA, the first molecule of acetyl-CoA will be converted into the first molecule of acetyl-CoA that is entering into the fatty acid biosynthetic pathway, will be entering into the stage 2, which is the carboxylation reaction. So that a single molecule of acetyl, only as the first molecule of acetyl-CoA is undergoing carboxylation reaction, the following acetyl-CoA molecule simply enters into the biosynthetic pathways, right? So the first acetyl-CoA that is entering into the de novo pathway will be entering next into the carboxylation step in order to make the reaction thermodynamically favorable, right? So now the 2 carbon acetyl-CoA, what happens when 2 carbon acetyl-CoA condenses with 1 molecule of carbon dioxide or 1 molecule of bicarbonate. The carbon dioxide is coming in the form of bicarbonate ions. So it undergoes carboxylation reaction. So the enzyme is the substrate as acetyl-CoA. So acetyl-CoA is undergoing carboxylation reaction. So what could be the enzyme? The enzyme is acetyl-CoA carboxylase. Since the carbon dioxide is coming in the form of bicarbonate ions, we have already studied in the carbohydrate metabolism. Whenever the carbon dioxide is entering in the form of bicarbonate, the coenzyme required is the biotin. So biotin is a must for a reaction because the carbon dioxide is coming in the form of bicarbonate ions. So the bicarbonate ions delivers the carbon dioxide towards the acetyl-CoA in presence of acetyl-CoA carboxylase enzyme with biotin as a cofactor, resulting in the 3-carbon moiety, the 3-carbon compound known as malonyl CoA. So this is the first enzyme, which is a very important enzyme which is involved in fatty acid biosynthesis or in the de novo pathway of fatty acids, acetyl-CoA carboxylase. Since it's a highly unfavorable reaction, thermodynamically it's a highly unfavorable reaction. This reaction is one of the regulatory enzymes of the de novo pathway. Clear? So this is acetyl-CoA carboxylase is a very important step. The carboxylation reaction of acetyl-CoA is very important because it converts the acetyl-CoA to a more favorable condition by converting itself into malonyl CoA. Again, acetyl CoA carboxylase or this malonyl CoA thus formed is not having any other fate in the body. Malonyl CoA, if malonyl CoA is synthesized in the body, it is solely diverted into fatty acid biosynthesis. Hence, the formation of malonyl CoA from acetyl CoA through acetyl CoA carboxylase forms or acts as the committed step of the reaction, because once malonyl CoA is formed through acetyl CoA, or once malonyl CoA is formed in the body, it will be focused only into the fatty acid biosynthesis. Hence, it is a committed step of the reaction. Clear? So that forms the stage 2 of the de novo pathway. Stage 1 is the translocation of acetyl CoA from the mitochondria into the cytosol through the malate shuttle. The second stage is the acetyl CoA thus entering into the cytoplasm is undergoing the first acetyl CoA molecule, because it's a series of synthetic pathway, or the 2 carbon moieties are getting added up in a stepwise manner, right, which is the reversal of beta oxidation. But the first acetyl CoA that is entering into the de novo pathway will be having an additional step, which is a stage 2 of fatty acid biosynthesis, which is a carboxylation reaction carried out by acetyl CoA carboxylase enzyme in presence of biotin. Clear? So, it is the initial and the regulatory step in fatty acid biosynthesis. It's a multi-enzyme complex having biotin as a cofactor, then biotin carboxylase, biotin carboxyl carrier protein, trans carboxylase, and a regulatory allostericity site. You just learned that acetyl CoA carboxylase is one of the regulatory enzymes of fatty acid biosynthesis. Okay, so it's a very important step in the reaction. So once the acetyl CoA is formed, the acetyl CoA is undergoing carboxylation to give rise to malonyl CoA. This malonyl CoA is then entering into the third stage of the fatty acid biosynthesis, that is the de novo pathway. So only when the malonyl CoA is synthesized, the first molecule of acetyl CoA has been converted into malonyl CoA, then only the de novo pathway will be initiated. So the priming steps of the fatty acid biosynthesis are: first is the translocation of acetyl CoA from the mitochondria into the cytoplasm, followed by the carboxylation of the first molecule of acetyl CoA to give rise to malonyl CoA. Now, once the malonyl CoA is formed, it is committed for once malonyl CoA acetyl CoA it is having many other fates, like acetyl CoA can be entering into TCA cycle or can be catalyzed into ketone body metabolism and all. And if the body is in demand of fatty acids, or if there is excess of carbohydrates and proteins getting accumulated in the body, then this acetyl CoA molecules thus formed from these carbohydrates and proteins will be committed for getting converted into fatty acids. So how can the fatty acid biosynthesis be initiated? The acetyl CoA will be converted into the malonyl CoA, and once the malonyl CoA is formed, it is not having any other fates in the body. It is not responsible for carrying out any other reactions. Once malonyl CoA is formed, malonyl CoA is responsible for initiating the biosynthesis of fatty acids, or the de novo pathway. Right. So once the malonyl CoA is, now we are having a malonyl CoA with us. Now this malonyl CoA initiates the de novo pathway. Right. So before entering into the de novo pathway, we should know what is the enzyme involved in the de novo synthesis or fatty acid synthesis. The fatty acid synthesis is again catalyzed by a multi-enzyme complex. We call it as FAS, that is fatty acid synthase complex. So what are the enzymes involved in the fatty acid synthase complex, or how does it appear? How are they arranged? Each enzymes, how are they arranged? And how do they bring about the fatty acid biosynthesis that we are going to study? Okay, so the fatty acid synthesis, the de novo pathway, involves 3 stages. First is the translocation of acetyl CoA from the mitochondria to cytoplasm. Second is the carboxylation of acetyl CoA, which forms a committed step of the reaction. That is, acetyl CoA undergoes carboxylation in presence of acetyl CoA carboxylase, and this acetyl CoA carboxylase in presence of biotin converts the acetyl CoA to its next higher member, which is known as a malonyl CoA. Once the malonyl CoA is formed, the acetyl CoA cannot be deviated to any other pathways. It cannot enter into TCA cycle or ketone body metabolism. It will be directly, it is then funneled into the fatty acid biosynthesis. Clear. So this forms the committed step of the reaction.